Hello friends, and welcome to our video lecture on topic C1.2 Cell Respiration. Our guiding questions: what are the roles of hydrogen and oxygen in the release of energy in cells, and how is energy distributed and used inside cells? Since cell respiration is all about making lots and lots of ATP, we're going to look at the life processes that require all of that ATP. We're also going to look at the structure and function of ATP. We're going to distinguish amongst ventilation and gas exchange and cellular respiration. In bio, these are all very distinct, related, but distinct processes. Outside of bio, sometimes we mash them all up into respiration. We'll talk about the differences amongst these here and why that's important in bio. We're going to also distinguish between aerobic and anaerobic respiration, and we will wrap up with a little look at methods for measuring rates of respiration. So there are three main categories of processes that require lots and lots of ATP. The first one is anabolism. Remember that anabolism is the piece of metabolism that leads to the production of macromolecules. We're taking small things and gluing them together to make them big things. This is a condensation reaction, also known as dehydration synthesis outside of IB world. Here we've got our glycine molecule. Glycine is an example of an amino acid. Here I have another glycine, another amino acid, and we know that if we put two amino acids together, we end up with a dipeptide. We're going to use the energy that is stored in the chemical bonds of this phosphate group to be able to form that peptide bond between those two amino acids. Here we have phosphorylated. That means simply that we added a phosphate group to that first glycine, that first amino acid, and that energy transfer that comes with that phosphate group transfer allows us to build that peptide bond between those two amino acids. Then that phosphate group pops back off. We can use it later in cell respiration, glue it back onto this ADP to make more ATP. Another cell process that requires ATP, this guy right here, is active transport. Remember that active transport is the movement of molecules against their concentration gradient. So I'm going from low concentration to high concentration. We're trying to hoard these molecules together. The universe is not stable when we hoard things, so we need energy to make it happen. And here we see we have some ATP. We are going to chop off that phosphate group again. That phosphate group helps us to transfer that energy to our protein pump here. The protein pump can use the energy from that phosphorylation, the movement of that phosphate group, to push those molecules against their concentration gradient. The last chunk of processes that we're going to look at here is movement. And we have lots of movement. We can be talking about like muscles moving, that requires lots of ATP. But we can also be talking about endocytosis and exocytosis, the movement of vesicles into and out of cells. We can be talking about moving chromosomes around during mitosis. Lots and lots of ATP is required to make all of this happen. So what even is ATP? Adenosine triphosphate. It's this molecule right here, which should look a whole lot like the nucleotides that we studied when we were looking at the structure of DNA and RNA. ATP is in fact a nucleotide. What's a little bit different from this guy compared to the monomers that we talked about when we were looking at DNA and RNA is that instead of one phosphate group, we now have 1, 2, 3 phosphate groups. 3 is tri, and this is why it's ATP, adenosine triphosphate group, 1, 2, 3 phosphate groups. We still have our ribose sugar. We still have our nitrogenous base here. This one happens to be adenosine. ATP, adenosine triphosphate. Its job is to take this last phosphate group. This phosphate group here can pop off of the ATP and be transferred to something else. We call that process phosphorylation. Phosphorylation. What's pretty cool is when we transfer that phosphate group, we also transfer lots and lots of energy that is stored in the chemical bonds. And so we can transfer energy to molecules like enzymes or muscle fibers or protein pumps through this process of phosphorylation. We call this phosphate group all by itself an inorganic phosphate, or PI. Well, that's a P. Inorganic phosphate group. What's pretty cool about cellular respiration is that I can actually glue that phosphate group back on to that nucleotide turn it back into ATP. So cellular respiration is used to replenish our supplies of ATP. We're going to glue together that inorganic phosphate group and this molecule that only has two is di. We're going to call this ADP because there's only 2 phosphate groups instead of 3. Photosynthesis can also glue together these inorganic phosphate groups and ADP. And one final process, chemosynthesis, which is a lot like photosynthesis, except instead of using light energy, we're going to use chemical energy to glue together that inorganic phosphate and the ADP to make lots and lots of ATP. And then once I have that ATP, we can dephosphorylate it. Oh, is this going to fit on the screen? It did. We can dephosphorylate, pop off that phosphate group, turn our ATP back into ADP and inorganic phosphate, and then respiration or photosynthesis or chemosynthesis can glue those guys back together to make more ATP, and this process can just happen over and over and over again. What's crazy cool, we humans need to use about 120 moles of ATP molecules every single day. Remember that a mole is 6.02 times 10 to the 23rd molecules of ATP. So we're talking about a lot of molecules of ATP every single day. But what's cool is that we only have about 0.2 of a mole of ATP in our body at any given point, which means that this process of dephosphorylation and then rephosphorylation, this process of ATP to ADP back to ATP has to happen just about 600 times for every single one of our ATP molecules every single day. That's crazy. That's the job of cell respiration. What's interesting, though, is that this process is not 100% efficient. A little bit of heat is propagated every single time we carry out these chemical reactions. A little bit of heat is lost to the environment. And so we need molecules like glucose or lipids in order to replenish that energy that is lost in these phosphorylation, dephosphorylation processes. How cool is this, though? It's happening in your body, every single one of those molecules, 600 times a day. Pop off the phosphate, pop on the phosphate, pop off the phosphate, pop on the phosphate. So sometimes outside of bio class, we use the word respiration to talk about breathing. In bio, we need to make sure that we are keeping cell respiration distinct from this idea of just breathing. So breathing or ventilation, pushing air into and out of our lungs, is important so that we can carry out gas exchange. Gas exchange is getting O2 into our blood and CO2 out of our blood so that we can carry out cell respiration, using that oxygen so that we can make lots and lots of ATP super efficiently. Cell respiration is using glucose and other molecules to produce lots and lots of ATP. Respiration for us in bio world is just this process that's happening in our cells. It's not ventilation, which is moving air in and out of our lungs. It's not gas exchange, which is trading carbon dioxide for oxygen. So just be super careful. The definition of respiration, releasing energy from carbon compounds like glucose to make lots and lots of ATP. That's just this chunk. It's not the breathing chunk. Keep those separate in your brains. Focusing here just on cellular respiration, we have 2 pathways that we can take to make our ATP. Aerobic requires some oxygen. The anaerobic pathway, the an for no oxygen is required. Both of these can start with some glucose. We're going to break that glucose down. When we break the glucose down, we're going to have some energy released in those chemical bonds. We can use that energy to glue our phosphate groups back onto our ADP to make lots and lots of ATP. In aerobic respiration, we're talking about 30 plus molecules of ATP per every 1 glucose that we start with. That's a lot of ATP. That's pretty fabulous. In fermentation, our anaerobic processes, we're talking about really only just 2 ATP molecules for every 1 glucose that we start with. You might be thinking, why even bother them? This is so much less. This clearly is way better. Why do we even have anaerobic respiration at all? So there is an evolutionary benefit to us having this backup plan. Sometimes our ancestors a long, long time ago had to run away from predators really fast. Or if they were really, really hungry, they may have needed a spurt of energy to catch a prey. This fermentation process, this anaerobic process of making some ATP, provided ATP even when we were pushing our bodies faster than we could get the oxygen to our mitochondria. Nowadays, it's really more of an athlete exercise kind of backup plan. So when sprinters push their bodies a little bit past what we would consider normal, their bodies can go into this fermentation process, make this ATP super fast, but just for a very short amount of time. Good intense exercise, but just in bursts. What's pretty cool about this process, we end up with lactate instead of CO2 and water like we do at the end of aerobic respiration. That lactate is then sent to the liver for processing. Sometimes that lactate gets built up in our muscles a little bit, and that's what makes our muscles sore. A little bit more comparing and contrasting of aerobic versus anaerobic processes for producing ATP. Aerobic, again, means that oxygen is required. Here we have that oxygen, an example of a word equation to describe aerobic respiration. We've got glucose and oxygen producing the waste products of carbon dioxide and water. And of course, lots of ATP is produced as a side product. We can start with glucose, which is a carbohydrate, but aerobic respiration is a little bit more flexible. We could also use a lipid or a protein to begin the process. Hopefully, though, we're not using the proteins. The hydrolysis of proteins to produce ATP means that we're starving, and that's bad. So we can use proteins, but let's not. 30 plus ATP molecules are produced for every one glucose that we start with. That's a lot of glucose. That's a good thing. Aerobic respiration begins in the cytoplasm, a process called glycolysis. We'll talk about it later. It's going to finish in the mitochondria, the mighty mitochondria, powerhouse of the cell. I don't actually know what that even means, powerhouse of the cell. What I do know is that mitochondria has lots of super cool compartments, and one of the benefits of compartments in an organelle like the mitochondria is that we can set up different chemical environments to carry out different processes at different rates. We have this one compartment here known as the intermembrane space. We have this other compartment here known as the matrix. And in our next lecture, where we look at a little bit more detail at these processes of aerobic respiration, we'll talk about the benefits of those compartments and the chemical environments that get set up in those different spaces. In our anaerobic processes to produce ATP, of course, no oxygen is required. That's the an in anaerobic. In us humans, other animals, some bacteria, that glucose gets broken down into lactate. That molecule lactate can sometimes build up in our muscles and make our muscles sore. Eventually, it gets pushed out to our liver for processing. In super cool organisms like yeast, what happens instead of producing lactate, that glucose is converted into ethanol and carbon dioxide. If we're talking about baking bread and we threw some yeast into our bread, the carbon dioxide gas bubbles are what make our bread rise. So fluffy bread comes from yeast carrying out this fermentation process. If we're talking about yeast making some beer, the ethanol is what makes the beer. And so yeast can be used, fermentation processes of yeast can be used for both baking and brewing. A limitation of anaerobic processes for making ATP is that we can only start with carbs. No lipids are involved in anaerobic processes for making ATP. We only get 2 ATP molecules for every one glucose that we start with, but all of the steps occur in the cytoplasm, no mitochondria required. So again, in us humans, we have this anaerobic process, fermentation, that allows us to produce some ATP. And when we start to exercise, we do tend to breathe faster and faster. That helps us get rid of our CO2 and push some more oxygen into our bodies so that we can carry out more aerobic respiration, make lots and lots of ATP. However, this movement of oxygen doesn't happen as quickly as does our need for ATP. And so we have this little bit of space while our body is playing catch up to get that breathing rate increased. We have this little bit of space where I don't have quite enough oxygen getting moved to my mitochondria quickly enough to do aerobic respiration. And so here we have our anaerobic processes occurring. We're going to make some lactate and a little bit of ATP, because a little bit of ATP is better than no ATP. This results in what we call an oxygen deficit. This is the difference between how much oxygen we need and how much we're actually getting to our mitochondria. That deficit is then kind of transferred into what we call a debt, and this is the extra volume of oxygen gas that we need to breathe after we have finished our exercise in order to replenish and restore all of the processes that we had going on before we did our exercise. If you've ever gotten to the end of a race or a game and you're still heaving those big breaths, this is what's happening. This is our oxygen debt. What's happening with all of that oxygen? We're taking the lactate that we made in anaerobic respiration here. That lactate that we made, we're going to convert it into pyruvate in our muscles. Oxygen is needed to make that happen. We're also just going to replenish all the oxygen that we pulled off of our molecules of myoglobin in our muscle and other processes that need to just kind of get back to homeostasis. So those big breaths that we take at the end of exercise, that's us fixing our oxygen debt. And because in science we do love to investigate processes so we can learn more about them, we do love to know things in science, we have devised lots of ways to investigate the rates of respiration. Remember that respiration is the process of producing some ATP. So here we're talking about ATP production by this lovely grasshopper here. This is not the same as measuring the rate of breathing. We could also do that. We use a spirometer to measure the rate of breathing. This device is not a spirometer. It is a respirometer. Be careful, they sound a lot alike, but they are not the same. Respiration for us in bio world is not the same as ventilation. It's not the same as that rate of moving air in and out of our bodies. What this respirometer is doing. So we have our manometer here in the middle. This is going to measure some pressure changes. We have gas here, we have gas here. I have a living organism over here. This living organism is going to consume some oxygen to make some ATP. As that oxygen is being consumed, it's being pulled through this tube. We're going to have a decrease in pressure. That decrease in pressure is going to cause the colored water here to move up, and then I can measure with my graduations over time how far, how fast does this water move up the tube. The reason that we've got this CO2 absorbent here, this is probably some potassium hydroxide. When potassium hydroxide interacts with carbon dioxide, we end up with a molecule known as potassium hydrogen carbonate or potassium bicarbonate. This is a solid that doesn't take up any space in the chamber. If we didn't have this stuff, the CO2 absorbent, what would happen would be really not much of anything, because there would be equal amounts of oxygen coming into the grasshopper and CO2 going out of the grasshopper. So you wouldn't have any changes in volume leading to that change in pressure, leading to the movement of the water in the manometer. So this, again, potassium hydroxide or whatever other carbon dioxide absorbent that we've got in the chamber with our living organism just helps us to not have carbon dioxide gas taking up space in the chamber. Over here, I do have some object that gives me equal volumes to the stuff that I have over here because I don't want to have more space and less space. So this is just a control for volume. And again, as the grasshopper is consuming the oxygen, there's a decrease in pressure here, which allows the water, the colored water, to move up that tube. We can measure over time, because we want rates to be over time, the distance that that water moves, and that's our rate of aerobic respiration. We can also measure rates of anaerobic respiration or fermentation with a gas collection device. So here we've got maybe some yeast and some glucose and some nice warm water, and that yeast is going to carry out fermentation. One of the waste products of fermentation, of course, is CO2 gas bubbles. Those gas bubbles are going to go up and through the tube. And then here we have a graduated cylinder that's filled with water. Those carbon dioxide gas bubbles are going to move up through the water and collect here. And as the carbon dioxide collects, it's going to push the water level down, down, down. And so again, we can measure the movement of that water down the graduated cylinder as the carbon dioxide gas is collected at the top of the cylinder. We call this volume by displacement gas collection. Again, we can do this with anaerobic fermentation of yeast because that carbon dioxide gas is a waste product. And on that note, my friends, we have completed all of our objectives for today's lecture. We talked about those life processes that require ATP. There were three main categories. Those were active, transport, and anabolism, and also movement. We talked about the structure and function of ATP. It's all about transferring that phosphate group, phosphorylation, with lots of energy to the molecules that need that energy. We talked about how ventilation, moving air in and out of our bodies, is not the same as gas exchange, which is trading CO2 for O2, which is also not quite the same as respiration, using the O2 to make some ATP. These are all related. They need each other, but they are separate processes. We talked about how aerobic and anaerobic respiration fermentation processes are important. Both of them are good in different ways. And we talked about how we can use respirometers to measure the rates of respiration. Great work today, my friends.